Hello and welcome back to the channel. I'm Sachin and we are back with a new video. Today we're going to talk about motif finding and why do we need it. Let's get started. So first we're going to talk about LAC operon. We have 6 sites, a CAP site, promoter, operator, and then 3 other sites, LAC Z, LAC Y, and LAC A. RNA polymerase binds to the promoter, which then transcribes the next 3 sites. The thing is, now the gene is always on. We need to save resources. So we have something called as operator, which binds to a repressor. And when the repressor binds to the operator, there is no transcription, which means the gene is always off. But now what we need is a condition, because we need to turn the gene on and off. So if we have lactose, we also have something called as allolactose that binds to the repressor and changes its shape, which then means that the repressor will no longer bind to the operator and hence transcription will go on. Well, let's see if glucose is present. If glucose is present, we just break it down. But if it's not present, we have more of something called as CAMP. And we have more CAMP, which means CAMP binds to the activator, which increases the rate of transcription. CAMP is cyclic adenosine monophosphate. A similar example can be seen with TRP operon. Operator is where repressor binds, promoter is where the RNA polymerase binds, and then there are other sites. So if we're low in tryptophan, we make it. If there's high tryptophan levels, tryptophan attaches to the repressor, which attaches to the operator binding site, exactly as the previous example, and hence pausing transcription. Why do we need this, or why are we talking about this? Is that, let's say we have some of these CAP sites, or as we talked about in the previous video, eco R1 sites, usually similar. So what happens is that we need to find out the positions of these given these strings. How do we find that? Before that, let's look at a small little concept called a consensus string. This will also give you the idea of the definitions we're going to use. So let's say we have 7 sequences, S1 to S7, which will be generalized as SK, each of length L. The alignment matrix would have a shape K times N, where N is the length of the motif, right? So let's look at another small concept called the consensus string. We have 7 sequences right here, which will be generalized as SK, each of length L. The alignment matrix would have a shape K cross N, which will be K, here will be 7, and then N will be the length of the motif. So we have 56 characters here, and then we have a profile which will give us a consensus string based on what is the maximum numbers of the current nucleotide in the current position. But now the question is, how do we find these start points? One of them answers is brute force. So let's say we have these start points, and these are the final points we need to find. The toy problem here has 4 sequences, 9, the string length is 9, and the motif length is 3, right here. And let's say these are the solutions. What we need to do is that take all the starting points, find all the sequences, all the possible sequences, and then make permutations of each of them. So the permutations would be something like this. So we'd have this start point, this start point, this start point, and this start point for the first one. And then we go 1, 2, 1, 1, 1, 3, 1, 1, 1, 4. And then similarly, once we're done with this, we go 1, 1, 2, 1. And then we keep going until we find all of these. Then we construct a profile for each of them like we did here. And then we score each of them. But there are different ways of scoring. The one way of scoring is, which we'll come to very soon, and then we pick one with the highest score. How to score them? So we sum the value corresponding to the consensus base for all the positions of the motif. Highest score would be K times N, in which all the motifs match, or we'll have the exact match in them, which is highly unlikely. And then the lowest score would be K cross N, or K times N by 4, where none of them match and they're just randomly distributed. All of them have equal probability of being present. The thing is, this is quite difficult for the computer, also time consuming, because as and when the strings increase, or also the number of strings in a motif and the length increases, it becomes exponentially more tiring for the computer to actually find it, which will in turn take more time. The question is there a smarter way? And yes, there is. The algorithm is MEME, or just in short, expectation maximization. Here we have the same example from the previous slide. We have length of the string 9, we have the width of the motif 3. What we do is we have random start points picked up, and then we use those to get the string in the sequences, and with those we make a PWM and we score it using log likelihood. Then what we do next is you take every starting point and score them according to this PWM, and whichever has the highest Z scores is actually taken as the start point for the new matrix. Then a new PWM matrix is made using those start points, and then, as you can say, we repeat these steps until the log likelihood of the current actually goes down under a certain threshold, and then we return the most common Z here is these, and then we return the most common Z. Why does this work? Because the more number of motifs match, the higher the PWM score will go, which in turn makes it more likely to pick the repeated motif. So what's happening is we have this search space. This is the point we want to actually be. This is where we started, but what will happen with this is that we'll, at best, we'll go to what's being repeated right here. So what we want to do is we want to do something of this sort. We will have multiple start points, and then this point right here will be reached more than the others, because these will just go to their local maxima. But if we take large enough samples, we will get to the global maxima more number of times than we actually touch the local maxima. There's also a paper linked in description. It's a study about some of the motif finding methods that you can find. If you liked the video, please like, share and subscribe. Thank you and have a nice day. See you.